Thank you, Ingrid, for the introduction. So today I'll be talking about harnessing gene editing to develop climate-responsive oats. So I want to start with the question, how many of you guys consume oatmeal in your breakfast? Okay, great. How many of you have eaten like energy bars that has oat in it? I'm sure you might be using beauty products made of oats as well. Okay, perfect. So it's an important cereal, right? So it's a versatile cereal that's grown as a fodder and silage. It's rich in beta-glucan, which is a soluble dietary fiber. It's good for your heart health and your overall well-being. And then it has several natural antioxidants, which are also good for your overall health. And also it's the largest exporter of oat is Canada, so it's also contributing to the Canadian economy. So that's how, in terms of health benefits and Canadian economy, oats is really important. That's why we're working on it. And moving on, it's a healthy cereal. It contains like 60% of carbohydrates as starch, and it has balanced amino acid profile for proteins, that's 15%, and then lipids, and then beta-glucan can range up to 6%, which is highest in all the small grain cereals. So what's the oat struggle? Why is it struggling in the Canadian agroclimatic conditions? So once the oat is sown in around early May, so this is the seedling. So how many of you have seen the oats flower? Is it a pink? Like this? Does it have like a pink color on the flower? Do you agree on this? Does it look like oat? Oh, I mean it's still beautiful, but it's green in color. It's not a pink flower. But still, that's how it looks when it flowers, and then it matures. However, until it reaches the time of maturity due to climate change, there is a problem. So it can be amenable to winter storms and crop lodging like this, which is not desirable. The farmer loses the whole production. So what farmers generally do, they spray this glyphosate Roundup chemical, which is a pre-harvest desiccant, and it promotes the plant to maturity. But this is not desirable because it will leave the footprints, right? Like you don't want to have the chemical footprints in your oatmeal that you consume or the beauty products that you apply. And it's also not acceptable by different agribusiness industrial people who take up oats and then put it in the supply chain. So people, they don't want to have the chemical treated oats. So that's why what we are doing is we want to work on the flowering time, and we will be using gene editing to alter the flowering time and maturity. So we want to play with the oats maturity and flowering time without chemicals, but by modulating the gene that controls flowering time. So that's my long-term goal, and the project goal is to develop high-yielding spring cultivars which are adaptable to short Canadian agroclimatic conditions. So we'll be playing with the gene that is responsible for the flowering time. That's how we can expedite or alter the oats flowering, which is suitable to different agroclimatic conditions, which is changing as well. But oat, it's very complex. It's a large genome. It has like 3 subgenomes and 1 nuclei, and then it has inversions and translocations. I'm not going into a detail, but it's a complete mess. You can see it here. So there's a lot of rearrangements going on. So it's not easy to manipulate oat at the genetic level. From this picture, you can see it. But what's known about the oats inflorescence? So this is the oat flower as I showed you before. So this is the gene that is VRN3 that's located on the oat chromosome, and this delays the flowering or time to flower by 5 days, which is a significant number because you can reduce the flowering time, or I'm not saying reduce it, so adapt it to whatever the conditions you have. So by playing with this gene, you can create variation in terms of flowering time in oat by tweaking the gene, this one. So we want to use gene editing, which is a CRISPR-Cas9 system, to modulate this gene that is VRN3, which is associated with flowering time and yield. So generally what happens when you try to reduce the flowering time, the yield is also, there's a yield penalty. So if we don't want to reduce the yield, but still, we have to play with the flowering time, the yield is a really important factor. So before moving forward, I'll talk brief about the CRISPR-Cas9 system. So it consists of a Cas9 protein right here, and then there's a guide RNA. This is the target sequence. This is our gene which we are targeting. So once this is delivered in the plant genome, it forms a complex that has the protein and the guide RNA, which is the target gene sequence. Then it goes in the whole oat genome and does the surveillance, and then when it finds the target gene, that is VRN3, this binds to the gene. And then there's a PAM site, so you know there are 4 nucleotides, A, T, G, C. So there is a PAM region for how we design the guide, so N can be any nucleotide, and then there is GG. So this is the region where the 2 to 3 base per upstream of this region, the Cas9 protein will cut the DNA. So when it cuts the DNA, there is a double-stranded break, which is lethal for the cell. So cell will definitely try to repair this. That's what we want. So when it repairs it, it creates an error, which leads to mutation, which will lead to altering the gene function. Whereas there is another approach where you can put your donor of your own interest if you want to put any other gene or any other target sequence, any of those ATGCs you want to put, you can also put your own donor sequence. But we are more interested in this error prone, so we want to make a change and let the DNA make the mutation. So how we do it in a lab? So we have oat seeds, we make callus out of it, it's like stem cells, so it can give rise to the whole plant. So you can regenerate it afterwards. And then you put it on osmotic media, so it's a sugar-rich media. So with reverse osmosis, it just opens up the cell wall, because cell wall is very rigid and it won't let your the CRISPR-Cas9 system to enter the plant genome. So we will deliberately put it on a sugar-rich media that opens up the cell wall. And then this is the CRISPR construct that I was talking about before, the whole assembly that has your guide RNA, has your Cas9 gene that will encode for the Cas9 protein, and it also has a selection marker which will be used further. So once you bombard or use helium gun bombardment, or there is agrobacterium, there are different methods of plant transformation. So we use one of those to put this construct into the oat genome. And you can see here some of these calli which are dying. That means they don't have this construct, because this construct, it has a selectable marker gene. So once this construct goes into the plant genome, it will survive on that selection media. So the ones that are surviving, that means they have this construct. So we are successfully transformed the oat genome, but then the picture starts for further, we do a shoot regeneration, we put it on a rooting media, then we produce roots, and then that's how we end up getting these transgenic plants. So once you have these transgenic plants with all those gene alteration, you will sequence your gene, because you know the gene sequence, you will amplify it using a polymerase chain reaction, which is PCR. I hope you guys might be aware of it. And then you look for differences in flowering time. That's what our major goal is, right? So here I'm really proud to share, we are like first successful milestone towards the punctual oats, because we want to adapt it to the changing agroclimatic conditions. Our lab has successfully produced gene editing in this gene. So you can see there is a deletion of 4 base pairs, and then this is a wild type substitution, like there is an addition of nucleotide T, and there is a deletion of 4 base pairs here. So you're assuming that this change in the gene sequence will change the flowering time, which we'll be screening in the next generation. And also, so we are doing this so that you can continue to experience the warmth of this delectable oatmeal bowl that a lot of you guys, I see you consume in the morning, and we won't let this snowstorms ruin the oat plant so that you're not able to consume your favorite breakfast. And at last, I would like to thank my supervisor, Dr. Singh, he's also here, and my committee members, their collaborators from Agriculture Food Canada, who are helping us with the phenotyping and also finding the gene and working forward on that. And all my lab members, some of them are here at the talk, and also our funding agencies, which are really important, that fund our project so that we can work on it. And that's it, and thank you. This is the Singh Lab website, and this is you all guys chilling during this summer barbecue at Dr. Singh's place. Thank you so much.